Next talk is from Arinjay. All of you know him. So he's a PI at the Saskatchewan University in Canada. And above that, he's also co-lead of the One Health program in Canada. And he's also one of our ECR representatives. So I guess if you have anything, go talk to him. He will be thrilled to have a talk about science and what else. And today he's going to talk about coronavirus's interaction and the immunological consequence of those. So you can go.
Thank you very much. Good morning, everybody. I'm very grateful for the invitation. Maybe I'm not audible. There you go. Very grateful for the invitation, and thanks UKICN for sponsoring the travel as well. I don't have a lot of time, but what I was hoping to do today is tell you about two short studies, and I'll show you only one data slide, and we can chat over lunch and perhaps coffee. I live and work on Treaty 6 territory in Canada, which is the homeland of the Métis, and I'm a settler in Canada, so I'm extremely privileged. All the data I show you today is coming from the hard work of grad students, postdocs, and undergrads who trusted a new PI 2 years ago. So I'm extremely grateful that we've got a fantastic team that's always working very hard. In fact, Victoria came to us from the Roslin Institute. And I remember, Paul, you wrote a letter of reference for Victoria. And she's doing some very good work, and you'll see a slide. Also very grateful to all our funding agencies that believe in our science. So we are located in the province of Saskatchewan, and we have the largest high containment lab in Canada. To give you an example in terms of size, this is a football field that's inside of CL3 containment. So we can house 1,000 pigs in containment for a large clinical trial. We've got bisons in containment, but we do work with bovine tuberculosis. Of course, bats, our favorite, and we can do all sorts of small mammalian animal models as well. So we are allowed to house any exotic animal species, any species you want to work with. We haven't tried polar bears or elephants, but I'm sure we can fit those as well. You see, we've got alpacas that are models for MERS coronavirus, bisons, of course, we've got bats, and we've got pigs and deer for chronic wasting disease, and moving on, deer for SARS-CoV-2 trials as well. With COVID-19, the federal government invested $200 million into VIDO, where we've now upgraded a portion of our CL3 lab into containment level 4 or BSL4. And this should go hot 2025. We're currently being inspected by the feds. We're also building a new animal facility that'll have year-round bat colonies, at least 2 bat colonies, and non-human primate colonies as well. We are renovating the old wing, and most importantly for VIDO, we've just finished manufacturing a vaccine development facility where we can make 40 million doses of GMP-grade human and animal vaccines. So I'm sharing this, so if you have opportunities to collaborate or sit down and have a chat with you, I'd love to do that as well. But coming down to my lab, I started my lab in 2021 when I moved from McMaster University and the University of Toronto. And research within my lab is focused on 3 themes. We are very interested in identifying how coronaviruses interact with their reservoir hosts. And our reservoir host model are bats. We're also extremely interested in studying how current and emerging coronaviruses interact with humans. We do in vitro models, we do ex vivo models, we've got organoids. And finally, research within themes 1 and 2 have now recently started informing theme 3 within my lab, where we are trying to develop next generation broadly protective vaccines and therapeutics. But I think today I'll focus on theme 1 because this audience is perhaps most interested in theme 1 today. So why bats? I started studying bats in 2013, and this is precisely the slide that my former PhD supervisor, who is now a professor emeritus, showed me in Berlin when I was visiting for a summer school. And if you look at bats and if you look at some of these viruses, for example, filoviruses like Marburg and Ebola, coronaviruses that cause diseases like SARS, Middle East Respiratory Syndrome, porcine epidemic diarrhea, swine acute diarrhea syndrome, and of course, COVID-19, paramyxoviruses such as Nipah and Hendra that cause severe encephalitis. And if you look at lysoviruses, a classical example of that being rabies, with the exception of rabies, none of these viruses in experimentally infected or naturally infected bats cause clinical signs of disease. Now, we don't define what a diseased bat would look like, but relative to animal models, bats are apparently healthy. And that's what intrigued me in 2013 to start studying the bat immune system. So the first thing I did, I walked up to my former supervisor and very excited first-year master's student, and I told him I'd like to study bats and viruses. And he asked me one simple question: what do you need to study viruses? And I was like, cells. And we didn't have those tools 10 years ago. So my master's project really was trying to develop these bat models so we could start studying bat viruses. So the first thing we did was we developed a Neptesicus fuscus kidney cell line, the 3 clonal populations, and then we also had to demonstrate that the cells were susceptible and permissive to viruses that are speculated to have evolved in bats. You can make cell lines, but if you can't infect them, you can't really study virus-host interactions. So at the time in 2014, we didn't have access to MERS, so we reached out to Dr. Vincent Munster at the Rocky Mountain Labs, and we went on to demonstrate that our cells are permissive to MERS-CoV, along with the control cells that we were testing, the Vero being African green monkey kidney cells, MRC5s are human lung fibroblasts. We also demonstrated a VSV, vesicular stomatitis virus, from the same family as rabies virus, could also infect the bat cells, and 2 of our clonal populations were also permissive to the porcine epidemic diarrhea coronavirus. We wanted to follow up on why the second population isn't, but we never had the time. And this is just a video of the bat cells propagating in culture. We do sell the cells through CareFast, and we do also share the cells with you. So jumping 10 years forward, over the last 10 years, we and our colleagues have shown, using all these bat cell lines, that bats really they seem to have evolved 2 key adaptations. One, they can control severe inflammation. So we see very little inflammatory cytokines when you infect these bat cells with viruses. And 2, we see a very robust interferon response, perhaps a response that can be modulated efficiently by viral proteins. So what we started doing in 2020, a lot of this work got shelved. So we're reviving it now since COVID research is on its way out, well, relatively speaking. And we started the study where we developed some primary lung cells. We have our established kidney cell line, and we took the most commonly studied human cell lines as well, and we infected them with ancestral SARS-CoV-2. Then we pursued proteomics and transcriptomics through a whole range of collaborators, including Dr. Sophie Marie Eicher, who's joining us as a postdoc. She's in the audience, and she can tell you more about the study. And Dr. Kaushal Baid in my lab, who's leading the study, along with Victoria, who's a PhD student, who's supporting the study. I'll only show you one slide because that's all the time I have. If you look at transcriptomics, so we're looking at RNA in these cells. In human cells, we do see a type 1 interferon response, which is your first line of defense when your cells get infected. And we're only looking at interferon beta transcript. It's one example of type 1 interferon. It does go up in human cells, Cal3 cells, but it's very variable. And we've shown this before in human patient samples in COVID-19 as well, that the response really varies between patients and between cell lines. However, in the bat cells, and we're showing you one cell line, we've got data from other bat cell lines as well, we see a robust increase of type 1 interferons. And we wanted to pursue this at the level of proteins. So proteomics is really new when it comes to applying the tools to bat cells. So what was funny was we could identify a lot of differentially expressed genes at the RNA level, but very few, only about 200 genes, were using proteomics. So perhaps that's a sensitivity issue, or maybe bats don't translate all the transcripts that bat cells are transcribing. This is a huge gap in the field. But we did see in human cells, IFIT1, that's a key interferon stimulated gene. It's an antiviral gene. In human cells, it goes up, but it's delayed. It goes up at 48 hours. In bat cells, in our hands, the basal levels are much higher than human cells, and it goes up much more rapidly in bat cells than in human cells. And we've done a lot of follow-up studies that I'll be happy to chat with you about. The second study and the last study I want to talk to you about is an in vivo challenge we did in the insectivorous bats with MERS coronavirus. This was a study that my colleague, Dr. Del Falzerano, formerly from Heinz Feldman's lab at the Rocky Mountain Lab, he started the study in bats. COVID broke, we shelved this project. We revived it last year, and we've started processing the data. So we challenged bats with MERS coronavirus. We went intranasal, oral, and IP, overkill of the virus. None of the bats got sick. Bats didn't lose weight. Bats gained weight during the course of the study. And fascinatingly, we saw at day 5 that bats do get infected in the lungs. Now, this is going to be very interesting, and we'll see how the field takes this. Because everybody in the field believes that coronaviruses have intestinal tropism in bats, which is not wrong. because disease ecologists have identified coronaviruses in bat intestines. But we believe that's perhaps a problem with the timing of sampling. In an acute infection model, we do see viral RNA very high levels in bat lungs. But by day 35, this infection's gone. It resolves it in the lung, but the RNA continues to persist in the bat intestine. We weren't able to isolate any live virus, but I think that's really key, that something's going on in these bats where the RNA can persist. And for whatever reason, over time, this can turn into infectious virus that's being shed. So when we publish this, we're trying to sell this as an animal model to study coronavirus persistence and factors that regulates persistence in bats. And finally, to touch upon, this question came up during the last talk, do all bat species carry zoonotic viruses? And we are very, very excited and intrigued with this question as well. We think bat species have evolved refined antiviral responses based off of the viruses they've co-evolved with. Not all bats carry all viruses. Not all bat species have the same adaptations. Bats are over 1,460 species. If you work with rodents, with mice, you really work with one species. You've got different strains, still one species. So imagine the diversity of immune responses and viruses that bats carry. And that's something we are very interested in, and we're actively pursuing this. We've got a nice paper in revision. And when that comes out, hopefully we'll be able to convince you that certain bats, especially rhinoceros bats, have very refined antiviral responses that allow them to control coronaviruses. So looking forward to the future, the direction we're headed for the next, I like to think next 40 years, I hope. Can bat viruses infect other species? And if we can identify these viruses, we want to be able to develop very novel cutting-edge diagnostics, therapies, and vaccines as well. And with that, thank you very much. Happy to take questions.
Is there any question?
It was really interesting to see the movement of the virus over time from the lungs through to other areas. And I saw very high samples in the brain as well. Do you have any opportunity yet to posit where the reservoirs might be within bats that carry this virus over long periods?
No idea. Absolutely no clue. So we think there could be individual level persistence of viruses, then there's population level persistence as well. And I don't have the tools to test population level persistence. But we are certainly interested if there are colleagues, perhaps not in this audience, but if there are colleagues that do disease ecology in the field, like Daniel Becker, who's a good colleague of mine over at Oklahoma University, University of Oklahoma, I think, or Oklahoma State University. We're trying to bridge this gap between field studies in bats and laboratory studies. We've always been disconnected. So hopefully that approach will help us start getting some of these answers to your questions.
I wonder if colleagues who work with mites might be able to identify whether the reservoirs lie in a second species.
It could be, and that's been done with Gabor Kimiensky's group, where they've shown that bat mites do carry a bat filovirus.
Hi, really interesting talk. I was wondering if you've looked at the sequence diversity of the viruses in your different cell lines and also the different tissues you've identified.
Yeah, so that study, Kaushal, the postdoc was leading it. He's writing it up now. We've also sequenced the virus over different passages, and we continue to see a loss of furin cleavage site. And I'm going to stop at that because that's going to raise a lot of questions, but we know why that's happening, and we think bats have a different endosomal route of entry, and they rely on that entry. It's not very furin dependent. So that's why vast majority of the viruses, sarbecoviruses that are being identified in bats, perhaps don't need a functional FCS. But that's our hypothesis, and hopefully we've got enough data to convince the reviewers soon.
Awesome, thank you.
Thank you. So Arinjay, lovely talk. I'm sort of melding it with what I've ever heard from Lin Far Wang in my head, right, on why he thinks bats tolerate infection. What body temperature are bats? And then when we think about like transmission from bats to humans, do they have to get over a hurdle of being effectively what we would have considered a temperature sensitive mutant?
It's a great point, Julian. So bats go through a wide variety of body temperatures, as low as 4 degrees when they're hibernating in Canada to 41 degrees when they're flying. And one of the hypotheses is that Tony Schkaunz had originally in the field is viruses have viral promoters that drive gene expression. Because bats have such a robust immune response or innate immune response, perhaps the viral promoters have evolved to be very potent in expressing proteins that can block those immune responses. When those viruses make that jump into humans, the viruses don't know that we don't have as robust of an innate immune response. They continue to pump those proteins that make us very sick very quickly. But none of us have been able to demonstrate that in the lab. We don't even have, I wouldn't know how to do it. But that's probably one of the key hypotheses that I'd like to test. If that's true, that bat-borne viruses have more potent promoters over long-term human-adapted coronaviruses.
I'd like to do a double act with Jules here. So Jules, correct me if I'm wrong, but in humans, we were seeing persistence of 3 or 4 sequences of viral RNA 32, 35 days out. And this was not just a little bit of RNA, this was pretty persistent signal. I don't think that that was just sort of residual RNA. I think that there's some cells with small amount of affection just continuing to produce that bit of viral gene. Do we need to change our traditional undergraduate understanding of virology? Could some cells be producing just some genes, and could therefore that be a mechanism for persistence and ultimate recombination? Because you've just said exactly what we saw in our humans. I'm going to pass over to Jules just to error correct me if I've got that wrong and to confirm. Over.
No, that's
right.
Yes. That's very cool because prior to the in vivo study, we published an in vitro study where we developed a persistence model in bat cells with MERS coronavirus. And we see 3 sets of populations, one that have low levels of RNA, one that have medium levels of RNA, and one that have very strong RNA expression. We could also detect proteins in the 3rd subset of cells. So there's certainly going on. We haven't gone as far as identifying specific cell types that allow for persistence, but our in vitro model works, and now we see it in vivo as well. At least at day 35, the bats seem to be able to persist with the viral RNA. We have no clue if that RNA can now give rise to replication-competent viruses, but that's probably our next step. We're going to stress these bats using different models to see if the bats can start reshedding the virus and ideally mimic what causes spillovers for bats.
Okay, thank you very much.